Good morning, students. How are you? Hope you all are fine. I, Kitakshi, teacher of Cambridge International School, Dasuya, today going to discuss a next topic, cell structure and its organization. But before start it, I want to discuss what we have done last lesson. So students, in previous lesson we discussed about cell and its introduction. In this, we have discussed about what is cell, who discovered cell, why cell is called basic unit of life. So beta, what is cell? Cell is a structural and functional unit of life. It is a basic unit of life because life arises from a single cell. Just a building is made up of bricks. Body of organism is made up of cell. About history, we discussed about who discovered cell first time. Robert Hooke in 1665, but the cell discovered by Robert Hooke, it was a dead cell. Then who discovered living cell? Leeuwenhoek in 1674. Cell consists of a nucleus which acts as a brain of cell. Who discovered nucleus? Robert Brown in 1831. Then inside cell a liquid present called protoplasm. Who discovered it? Purkinje in 1839. Then who gave certain features about cell called cell theory? Quine and Skradin. Who modified the cell theory? Virchow. This was all about history of the cell which we discussed in our previous lesson. Next, then we will discuss about why cell is a basic unit of life. As I already discussed, just a building is made up of bricks. Body of organism is made up of cell. Life arises from a single cell which combine together to form tissues. Tissues combine together to form organs. Organs combine together to form organism. Cell is called structural and functional unit of life. It is a very important question which we discussed in our previous lesson. Why cell is called structural unit of life? Because body of organism is made up of cell. Why it is called functional unit of life? Because all functions are performed by cell. Then we discussed gadgets to study cell is called microscope. There are two types of microscope which we use to study cell. One is light, one is electron. Light microscope having magnification range from 100 to 1500. Electron microscope having magnification range from 1 lakh to 5 lakh. And we also discussed about the structure of the microscope and its components. Then next we discuss about shape and size of cell. As you know, cells vary in shape and size. They may be oval, spherical, rectangular, spindle shape. Size of cell also varies in different organisms. Most of the cells are microscopic in size like RBCs, while some are large like nerve cell. Then we discussed about how can cells perform distinct function in organism. Cells are capable of performing multiple functions in an organism. Cell contains specific components called organelle. Each organelle in cell perform a different function. So this was all about which we have done previous lesson. Today I'm just going to discuss about next topic is regarding cell structure and organization. In this we will discuss only about plasma membrane in our today lesson. Next component we will discuss one by one in other lessons. Students, today I am just going to discuss about structural organization of cell. Cell is broadly formed of three major parts. These are plasma membrane, nucleus, cytoplasm with all cell organelles. One by one we will discuss all these parts in detail. First of all, today in this lesson, I'm just going to discuss only about plasma membrane. Plasma membrane. Now, what is this plasma membrane? Plasma membrane is a outermost layer of cell. It is a covering of cell. It separates the content of cell from external environment. Now, next is nucleus. Nucleus is the spherical body. It is present in the center of animal cell and is displaced to one side in plant cell. Nucleus is control center of all activities of cell. It controls all the activities of cell. It is a storehouse of information. It helps in the transmission of characters from parents to generation. Next is cytoplasm. Cytoplasm is a living substance of cell bounded by a plasma membrane called protoplasm. Protoplasm includes all components of cell except cell wall and content of vacuole. Now, protoplasm is further distinguished into two parts: cytoplasm, nucleoplasm. Cytoplasm is that part of protoplasm which occupies the space between plasma membrane and nuclear membrane. So, we will discuss all these components in detail. Cytoplasm further consists of subcellular components called cell organelles. One by one we will discuss all cell organelles in detail, but today this lesson I will just discuss about plasma membrane in detail. So dear students, here you can see clearly about structure of plant cell and animal cell. Plant cell consists of cell membrane. Animal cell is also surrounded by a cell membrane. But plant cell is different from animal cell. They are also having a cell wall present at the outermost. It is also outermost covering of cell. Here you can see cell membrane. What is the function of this cell membrane? Cell membrane, it just gives definite shape to cell. Now, next we will discuss about nucleus. Students, here you can see the nucleus, which is the spherical body present at the center in animal cell and displaced to one side in plant cell. Why it is displaced to one side in plant cell? Because the development of vacuole in plant cell. Nucleus is discovered by Robert Brown. Next my lesson, I will discuss all components of nucleus, its structure, its function in detail. Dear students, here you can see cytoplasm in plant cell and animal cell, which is present in both. Cytoplasm is a part of protoplasm. I already discussed with you. Now, what is this protoplasm? Living substance of cell bounded by a plasma membrane is called protoplasm. It is a jelly-like semi-fluid substance. Various cell organelles suspended in it. Protoplasm includes all the components of cell except cell wall and content of vacuole. It is further distinguished into two parts: cytoplasm, nucleoplasm. Cytoplasm is a part of protoplasm which occupies the space between plasma membrane and nuclear membrane. Cytoplasm contains 90% water, 7% proteins, 2% carbohydrates and lipids, 1% inorganic material, mineral, vitamins. Next lesson we'll discuss about cytoplasm in detail. Today I just only discussed in detail about plasma membrane. So now, what is this plasma membrane? Plasma membrane is the covering of cell that separates the contents of cell from its external environment. It is like the same as our body but protects our all internal organs of the body. Now next concept is regarding permeability, and this is very important. Permeability is the degree of diffusion of gases, liquid and dissolved substances through a membrane. Different type of membranes may be differentially permeable to different substance. Normally, permeability of a given membrane remains unchanged, but it may change with alteration in environmental conditions of the cell. Case of plasma membrane. Plasma membrane is selective permeable. What do you mean by selective permeable? Which allow only selective substance to pass through them. It is like a checkpost. They ask for license and your ID so that you can cross that checkpost. Same as plasma membrane, it just allow only selective substance to pass through them. And so this is a very important question, why plasma membrane is called selective permeable membrane, because it allow only selective substances to pass through them. On the basis of permeability, there are four types of membrane. First is permeable membrane. These membrane allow free passage of solvent, you can say water, and most of the dissolved substances. Example, cell wall in plant cell. Filter paper is made up of a pure cellulose. It also functions as permeable membrane. Next is impermeable membrane. This type of membranes with deposited of waxy substances like cutin do not allow entry of water, dissolved substances and gases. Here we can just take example of wall of cork cell or cuticle layer of leaf. Next is semi-permeable membrane. These membrane permits the movement of solvent molecule only through them, but prevent the movement of solute particles. Example, animal plasma membrane or egg membrane. Next is selectively or differentially permeable membrane, which is present in this type of membrane allows selective substances like passage of solutes along with solvent through them. Osmotic diffusion of water through selective permeable membrane starts from higher water potentials to lower water potential. Now next come to the functions of the plasma membrane. First, it binds the semi-fluid content of cell and give it a definite shape. Second, it functions as a mechanical barrier, protects the cell contents and maintains internal environment of the cell. Third, it is a selective permeable membrane. Being selective permeable, plasma membrane permits only certain substance to pass through them. Next, it is very flexible. Because of flexibility, plasma membrane enables the cell to engulf large molecules and food particles. Next, it defends against microbes. How? Because of chemicals present on the surface of plasma membrane help in recognition of foreign substances and defend against microbes. So all these are functions of plasma membrane. Now come to the structure of plasma membrane. All biological membranes are thin, film-like elastic membrane. Plasma membrane is flexible and made up of organic molecules called lipids and proteins. So it is also known as lipoproteinaceous in nature. Structure of plasma membrane can observed only through an electron microscope. Under the electron microscope, all biological membranes consist of three layers, two dark outer layers separated by a light middle layer. These corresponds to outer layer of protein and middle layer of phospholipids. Because of this membrane, plasma membrane is called as the unit membrane. Here you can see plasma membrane is made up of lipid sandwiched between two continuous layers of proteins. Here you can see a polar hydrophilic head group as considered as a lipid molecule. Center, these polar hydrophilic head group also contained a hydrophobic fatty acid tail inside and outside. These hydrophobic tails crowds inward away from the water. So here in structure you can see membrane is made up of phospholipid bilayer. Phospholipids are amphipathic molecules with a hydrophilic head and hydrophobic tail. These are attached to a glycerol molecules by a covalent bond. Now this is a very advanced model of plasma membrane called the fluid mosaic model of plasma membrane. Cell membranes are represented according to a fluid mosaic model due to fact that they are fluid, means the phospholipid bilayer is viscous and individual phospholipid can move. Mosaic means phospholipid bilayer is embedded with proteins, resulting in a mosaic of components. It was proposed by Singer and Nicholson. This model explained the structure of plasma membrane of animal cell as a mosaic of components, means lipid layer is embedded with different components such as phospholipids, proteins, cholesterol, carbohydrates. These components give a fluid character to a membrane. Each phospholipid has a hydrophilic head pointing outward and a hydrophilic tail inside. Plasma membrane has three types of proteins. One is integral proteins. These proteins form channel to allow the movement of large molecule and ion across the hydrophobic layer of the membrane. Next is peripheral protein. These are found embedded in a single leaflet of the membrane. They carry signal from one segment of membrane and relay it to another. Next is glycoproteins. They stabilize the membrane and responsible for intercellular communication. Here you can see a carbohydrate chain which is attached to a protein on outside membrane layer make a glycoprotein. About glycoprotein I already discussed with you. They stabilize the membrane and are responsible for intercellular communication. Then next you can see cholesterol. It helps the plasma membrane to retain its fluidity. It is present between phospholipids and prevent the compaction of hydrophilic tail at low temperature and their expansion at high temperature. So dear students, in the end of this lesson, I would like to say that first of all, you have to read the content given in NCERT book. For NCERT book, you can just click at the given below. Then you have to to understand the content given in NCERT, just go to this Diksha site. Here you will get a very relevant material and videos. Then try to understand the content, whatever I taught in my video. Try to understand the content given from these resources. Thank you.
If you're in a cell cytoplasm heading outside, your last stop's a barrier 8 nanometers wide. The cell membrane, our subject for today, it's as basic to life as DNA. It's selectively permeable like a border patrol of a country. It maintains control, selecting what leaves or what gains entry. It's a guard, bounce, a watchman, patrolman, or sentry. But not just a guard, it sends signals and nerves. White blood cells use their membranes to eat germs like hors d'oeuvres. Membranes have receptors, enzymes, and junctions. So what kind of structure can have all these functions? C-double-L-membrane, control and transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol. The fluid mosaic bilayer in us all. So let's take a look at what makes up the membrane. Phospholipids rule in this domain. In fact, membrane structure emerges directly from phospholipids' chemical properties. There's a head and a tail on every phospholipid. The tail's 2 long-chain fatty acids bound to a glycerol. It's made to order. The tail's non-polar, hydrophobic, fears water. The head's got a phosphate. It's charged negatively, makes the head hydrophilic, plays in water happily. So tail avoids water while the head's attracted to it. When phospholipids form the membrane, that's how they do it. Cause when phospholipids into water get submerged, a phospholipid bilayer structure will emerge. The tails hang together in a water-free zone. Hear the hydrophobic moan, wanna leave me alone. While the heads are sticking out, touching all those H2Os. Tails in, heads out. It's how every membrane goes. Tails in, heads out in a cellular sphere. It's the bilayered basis of membranes everywhere. C-double-L-membrane, control and transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol. The fluid mosaic bilayer in us all. But a cell membrane's not just a phospholipid scene. There's cholesterol, carbohydrates, lots of proteins. In fact, membrane proteins have a presence so great they often exceed the phospholipids by weight. And all of these components are in constant motion, moving, mixing like a boiling potion, flowing like dancers in a party in Mosaic. That's why the membrane's thought of as a fluid mosaic. Let's start with the proteins since they're key in this mix. Transmembrane proteins span the entire width. Typically their ports, think of channels of conveyors for things that don't go through the lipid bilayer. Any protein embedded in the hydrophobic middle built right into the inside is considered integral, while peripheral proteins either hang on the exterior or on the cytoplasmic interior. Cholesterol keeps membranes flowing with ease. In cold it keeps the lipids moving so they don't freeze. In heat it slows the lipids down, limiting their traveling. Cholesterol keeps membranes from unraveling. Membrane carbohydrates work as markers or signs, so your immune system knows which flag your cells are flying. The blood types ABO and B and A are about the carbohydrates on red blood cell membrane. C-double-L-membrane, control and transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol. The fluid mosaic bilayer in us all. C-double-L-membrane, control and transport, selective permeability. Phospholipids, carbohydrates, proteins, and cholesterol. The fluid mosaic bilayer in us all.